Welcome back to Offworld, the show where we talk about all things pop culture and space exploration. I'm Ariel Waldman.
And I'm Norm Chan. And we got a horror movie for you this time.
An amusing horror movie.
Yeah. This is the 2017 movie Life, which is set on the International Space Station, starring the prettiest astronauts you'll ever find.
And beautiful scenes and everything. It's an amusing horror movie is how I would put it.
I think when I first saw the trailer for this, it evoked two things. It evoked potentially gravity in terms of a quote-unquote realistic depiction of International Space Station.
Gravity the movie, not Gravity the physical sensation.
Yes. Microgravity, I guess, is what they were trying to simulate. And also Alien, because it is a organism that invades the space station. But the interesting thing with this organism is that it starts off very small.
Yes, yeah. So they get a Mars sample return. It docks with the International Space Station. They have some Mars dust. They put some water on it. And then they're like, let's expose it to an old Earth atmosphere. And suddenly it becomes alive and then starts growing quite a bit.
Mm-hmm. Growing stronger, more intelligent, and it quickly devolves into a horror chase sequence where the astronauts are picked off one at a time, just like in many other science fiction films. But the fact that the organism starts off as a microbe, as a single-celled organism, is why we have our guest today.
Yes. Yeah, we have Dr. Penny Boston from NASA Astrobiology Institute, and she looks at all sorts of weird little microbes and studies life here on Earth so that we can better understand how to find life in outer space or on other planetary bodies.
So curious what she has to think about this film. So let's jump to the conversation.
Today we have Dr. Penny Boston, the director of NASA Astrobiology Institute at NASA Ames Research Center in Silicon Valley, and an astrobiologist. Thanks so much for joining Norm and I.
Oh, I'm totally happy to be here chatting with you guys.
Yeah, so Life. Gosh, this movie was fun, I guess is a way of putting it.
It's fun.
So it's this movie that, they're doing a Mars sample return, and the Mars sample return arrives at the International Space Station, and, they're looking to see if there's any life at all in this Mars sample return. So, getting to that Mars sample return aspect, how would a Mars sample return work, for NASA or for any other agency? What's involved in actually doing a Mars sample return?
Well, really different from the movie, as you might guess. Many NASA groups and nationally convened bodies, labor mightily over how we do this. There are international treaties about how we do this, and we have to adhere to those because we're a signatory to those. And the crux of the matter is that we want to protect our home planet, no matter how remote the chance of some kind of misbehaving extraterrestrial life might be. And we also want to protect any indigenous life forms that we find on another planet like Mars or the icy moons in our solar system or anywhere that we can explore. So the level of containment that would be required and that are those processes are being planned and have been planned for many years are incredibly stringent. And we have a lot of plans now, but they haven't been entirely coalesced because this is something that you don't do in a big hurry. There's really a lot of complicated nuances. And the microbial world, which is really the size of organisms that we're contemplating, is something that we know only by assisted devices like microscopes and chemical sensing devices and so forth. So they're a kind of life that, while we're all related, and we have a lot of the same chemistry, their ways of doing things is, in many ways, a little bit more like chemistry. They're very tiny, so they can get everywhere. They experience their universe in a way that is much different than we do. So, for example, ordinary water, which to us is a very non-viscous, sort of thin fluid. If you're the size of a microbe, it's like swimming through molasses. You're small enough that you can be smacked around by the random Brownian motion of atoms and so forth. So their experience of the world is really different than ours, and so you're constantly forced to be rethinking how you relate to what the microbes could potentially do, how they could potentially get around.
I mean, of all the things you talked about, and there's so much to unpack there, I think the film touches on some of those, right? You have
Yes.
the CDC representative, played by Rebecca Ferguson, talking about all the signatories, people thought about the
Right.
protocols.
Yeah.
But then you also mentioned that something the film didn't consider, which is how you would treat the microbial life if you found it, because they have no concern for that.
No,
in the
apparently
film.
they're willing to lick it or stick it
Probe
up their
it.
nose or something.
Shock it with electricity.
Right, which is never considered a friendly act to any life form, I would think. No, the way that the containment is depicted is very far from what we are planning for. So we have a lot of complexities. So, the CDC aspect of it that they mention in the film has to do with the CDC's abilities to handle, study, and deal with exceedingly infectious life forms here on Earth. What they don't have to deal with is the degree of just chemical cleanliness that you have to deal with in a spacecraft, because instruments require very low background levels of interfering substances. And those can be just things from the air that are falling on the devices and so forth. So for planetary protection purposes, we worry about not only the organisms, but also the whole organic milieu that they bring with them. So for someone who's a geophysicist, maybe, who has a seismometer or some other device, a dead bug is a good bug because it's not interfering with your work. But from the point of view of life detection and assuring that we don't have interfering detections of life, and that we don't inadvertently allow possibly deleterious life to get out of containment, a dead bug is not a good bug because it's still there.
Yeah.
And its signal is still there. So, there are a lot of really finely divided components of how you have to approach this.
Well, and you mentioned planetary protection. For those people who don't know what planetary protection is, can you sort of
Yes,
explain
I
that?
can.
Because the thing I thought was interesting, if you can get into a bit, was the fact that the film focuses a lot on backwards contamination, which is another thing to sort of define.
Yes. So originally, in the 1950s, people started to worry about this. This isn't something that we just thought of yesterday. And an organization was formed internationally. It's known as COSPAR, and they have many aspects to what they do. One of them is to try to serve as an international channel for people to come together to discuss these issues. Originally, planetary protection was called planetary quarantine. And that was the idea that we would send our first humans to the moon, and we knew very much less about the moon at the time. And so the idea was to keep our first lunar astronauts and presumably cosmonauts, had they ever landed humans there, protected from the rest of our planet in case there was something on the moon. And as time went on, we got much more sophisticated in our understanding. The moon stopped being a concern, but then our eyes shifted to Mars and beyond. So you can break planetary protection down into two major camps. There is the forward contamination, and that is the concern that if there are life forms on another body, we do not want to contaminate that body and provide some sort of hazard to those indigenous life forms that live there. And for NASA particularly, we're concerned about mucking up the science because we now have increasingly high-resolution instrumentation to be able to figure out whether there's any little, extraterrestrial guys there. But that means that we also are at increasing risk of being able to detect and confuse our own microbes. So the protocol has to do with sterilizing or super cleaning of spacecraft and the instruments that go aboard them. So that's all forward contamination, away from our planet to some other place. And then the counterpart to that, of course, is been a very potent theme within a lot of science fiction, including the Andromeda Strain and many other movies, and that is bringing something back to Earth that might be a hazard to us here. And in general, I think that the consensus is that the risk is, or the probability, not the risk, the probability is very low. Because on this planet, pathogens tend to co-evolve with their hosts. They have to get smart, so to speak, at invading their hosts' defenses.
Which we see happen in the movie quite quickly.
Yes,
quite quickly.
And so we think that the probability is low, but the risk is significant, and the consequences are huge because this is the only home planet we have. And we're already doing a lot of things to this planet. We'd like to not introduce alien deleterious life forms. So that's how it breaks down.
For the film, to its credit, it doesn't bring life form to Earth, right? And the setting is the ISS. Right. Is that how a sample return would happen?
Many people have suggested either a lunar base or the ISS or some other facility. Now NASA, of course, is working on this gateway concept.
The lunar gateway.
The lunar gateway concept. If that comes to pass, that is a possibility, I guess. There have been many proposals for different places on Earth. There's been a proposal for a floating one, which I'm not sure
like
I'm
in
a
the
big
ocean?
Fan in the ocean because the ocean is a giant transport mechanism.
Yeah.
So I'm not really fond of that. Many places around the world have, contemplated whether or not they could safely house such a thing. We certainly know that the CDC in this country, and there are similar facilities in other developed nations, does a wonderful job of these outrageous pathogens like Ebola, Marburg virus, etc. But the stakes are very high, and also the public profile is very high. So
in the movie we see, so they've treated a Mars sample return to the International Space Station as being like Firewall 2 or something to keep it away from Earth.
Yeah.
And then in the movie, they talk about, it's gotten out. So the only way to contain it is to essentially get rid of the astronauts in the International Space Station altogether. And they
push
try
it away,
right.
Push
it away with a Soyuz, because that's totally possible. But
right. I mean, somehow, to me, this doesn't actually solve the issue. Not only do we not really want to push our astronauts and abandon them in space, but also talk about a clunky plan B. I mean, this is, you would have so many levels of containment, redundancies, triple backups, quadruple backups.
So that you don't have to use fire extinguishers on the International Space Station.
Or flamethrowers.
Right. Yeah, flamethrower. Yeah, that was what it was, a flamethrower.
I was particularly fond of the flamethrowers. And
yeah, so for people listening in.
I actually laughed out loud on the plane because I watched the movie on a plane going to Spain.
Yeah, which is, comforting. Yeah.
Right. It was the only place I could be trapped long enough to watch this movie. And the flamethrower stuff, well, I'm not sure.
Yeah, so in the movie, essentially, I mean, I call it a killer starfish is essentially what this little life form turns into, and then, it gets out of containment and it starts trying to murder people, and Ryan Reynolds has the great idea of, well, why don't we just put a flamethrower at it and burn it to death in the inside of the International Space Station
without knowing that you can actually burn it.
Yeah, exactly.
I mean,
and it turns out you can't.
You can't. Well, one of the things that really made me grit my teeth while I was enjoying an airline meal was the fact that early on in my career, when I was actually doing a NASA postdoctoral fellowship at NASA Langley Research Center back in the 80s, part of our project was looking at the behavior and capabilities of microorganisms in soils before and after major forest fires. So one of the things that was very clear was that there were a huge number of organisms that survived handily the burning process because they're so numerous and they're so tiny, it doesn't take much to protect them. And so you could capture soot now from the forest fires that are raging here in California, and you could recover quite a lot of microorganisms.
I think there's actually
It
some
actually disperses them, I think more.
I talked to some NASA interns the other day who I think said that they're doing just that. They're sending up these little weather balloons and collecting
that's
microbes
right.
from the soot in the air.
Yes, interns working for Dr. David Smith, right, who's looking at atmospheric microbiology and what microbes do in the air and stuff.
Very resilient, and even within the logic of the film, where they clearly state that this creature, every individual cell is intelligent and muscle and brain.
Right.
Burning would do nothing.
Right. All you needed was a failure of one entity.
Yeah.
Right. It's actually humorous that they used a starfish-like creature because there is a famous story of fishermen back maybe, I don't know, the 1800s, the early 1900s, not understanding that when you cut a starfish up, that you were not killing the starfish, you were essentially vegetatively propagating the starfish, and they would cut them up to try to kill them, to keep them away from, the important species, and they'd throw them all back, and so they were actually helping to propagate, huge numbers of the starfish. So I thought that was kind of humorous.
Maybe that's where they got their inspiration
Maybe it
from.
is, yeah, from that, because it's a well-known story, and ecological disasters that we bring on ourselves.
In terms of the research and containment, though, yes, the ISS is an interesting setting. Their version of the ISS has many more modules than our current one has. If we were to need to set up, the ISS as a research and containment facility for some type of extraterrestrial life, what would that lab look like? And did the way they depicted it in the film, is that an...
No. I think that the way it was depicted in the film was... containment light. So the way real extremely hazardous organisms are contained, there is a positive pressure that's brought to bear so that from the outside, so that there's no external leakiness. Well, the ISS, like all spacecraft that we've flown, has a leak rate. So the ISS is not particularly well suited for this in its current state. It would have to be significantly reworked, and I suspect that even if the ISS gets renewed for yet another cycle beyond 2024, we're still not looking at the time frame that we need it. But when getting back to your original question, the way the laboratory is set up, the positive pressure from the outside to keep anything from leaking. There are, levels of containment, not just wearing suits that completely cover the people, but they never, there's always firewalls of multiple sorts between the people and what could come out of the chamber. So, you would have to do many failure points within that system, whereas they had almost no margin, right? So,
yeah. Well, I mean, so the other question is, so again in the film, they get the Mars sample return, they get on the ISS. It's like kind of dirt. They essentially add water, starts growing. Simple.
Yeah.
but
I wish I thought that was going to be true.
Yeah. But would astronauts aboard in this scenario, aboard the International Space Station, or even astronauts if they were on the surface of Mars, have enough facilities to be able to pretty much confirm life on their own?
I think it would be very difficult. I mean, you look very skeptical. I know that look on your face. Yes. I often say that our current ability to detect life is many, many different ways that we bring to bear. So in my own work, and subsurface microorganisms in caves and mines, we often, even with all the laboratory facilities that we have, it has taken us many years to confirm that a lot of the materials that we are finding have life, because the amount of life per amount of stuff that you collect is very small, and many of the organisms are covered in minerals. So, and they don't grow very fast or do much that you can detect. It's not like, the microbiome that we're familiar with. If somebody takes a throat swab, they can tell within 24 hours or 48 hours if you have a strep infection or something. These are organisms that live on us, and they're used to big fat piles of nutrients, and they're all bloated and happy, love living on us and in us. But organisms in rocky environments, very slow to grow or do anything, very lean and very tiny. So, confirming them is an enormous labor. Yeah.
Yeah. So even with a ton of facilities and, let's say, international scientists, in the hundreds, you've done all of this interesting. You've gone deep into caves. You've discovered a bunch of stuff. How. So it sounds like it's pretty difficult to determine what is and isn't life sometimes.
It can be. The first specimens that we thought might be a living product were in Lechuguilla Cave in New Mexico, which is the deepest cave in North America and one of the biggest. And this is a gigantic maze caves, like 150 miles of map passage in this big cloud maze-like thing. And we saw this brown and orange and black and gray and all sorts of different colors, fluffy material. And frankly, it took us about 7 years to really demonstrate to ourselves, and then even longer to others, that we were actually looking at a microbial community and that they were covered in iron and manganese and that they were really alive and not just coated organic material lying around. And we've developed in our team and a few other folks that actually work in that area, we've developed sort of a experiential intuition that's really based on mineral properties. We see transformations of minerals and we go, I think little guys are at work there. But then you have to go through these many, many steps to really show that. And, at some point, I'm sure we'll have more sophisticated, tiny instrumentation that will be able to do all of those investigative processes, but we're not there yet.
I mean, to use your cave analogy, would it make more sense to bring the scientists to the location, to Mars in this example, than to bring small samples back?
That's a huge debate, yes. I mean, I come down personally on the side of human exploration because, first of all, I'm an explorer, and my entire career has, focused a lot on the human aspect. I'm particularly interested in our microbiomes and how they will, affect us. But I personally am a strong advocate for human exploration of suitable bodies, so obviously the moon and Mars. There is a huge trade-off. It's expensive to send humans. We're not quite ready for prime time. NASA's devoted to the idea of getting ready for that. And this new push back to the moon is viewed as another step in readying our species for getting to Mars. But in the meantime, you still want to be doing robotic missions, and you're going to be wanting to use robotic devices, even if we send humans there as we're planning and have them do the astrobiology because of this contamination issue. We are covered in stuff, and we have it all through our gut. And it's necessary to us. So it's not like we're going to have sterile, non-microbiome containing astronauts. They make lab mice that way, but it's not particularly good for the lab mice. And so we are going to have to deal with our own individual ecosystems that we take with us. And so for a potentially sensitive biological site, I think many of us envision that there will be sterilizable robotic and autonomous or semi-autonomous devices that can go to these very sensitive sites to perhaps sample.
And these sensitive sites on Mars are like, so they recently discovered there might be an underground lake on Mars.
Yes, they did. The jury is still out on that. There's been one report. And so I'm sure that all the folks that do the kind of work that led up to that claim in that paper are all busily trying to see whether they agree with that and find other ways to test it. If that turns out to be the case, then we do have wonderful analogs right here on Earth. Antarctica, which has these famous glacial lakes, as they're called. I think of them personally as fluid-filled caves with the ice as the bedrock. And so we have some limited experience with trying to deal with those. So that would be considered an extremely sensitive site because of should the presence of actual liquid water be confirmed under there. It would be incredibly alluring as a site to investigate. But of course, there's less spectacular but still fascinating examples like the recurring lines that appear, these recurring slope lineae that seem to be liquid coming out of different geological layers in crater walls or scarps. And there's considerable debate about whether that's really liquid. It's not easy to figure this stuff out from the kinds of instrumentation we still have. So they're a sensitive site. Presumably anything around the polar caps with the potential for fluid, underneath the ices would also be sensitive, and certainly caves would be sensitive.
Yeah, so I guess my final question is, let's imagine that we actually discover life definitively on Mars, either currently living because it's deep in a cave or something, or it lived 3 billion plus years ago. How do you think that would change society? How do you think that would change you personally?
Well, I will have achieved my lifetime dream of living long enough to see that happen. I mean, this has been my passion and love since I was 8 years old. And amazingly enough, I've managed to do it as a grown-up for a living. So for me personally, it would be just unbelievable. I'm not quite sure how. Maybe I would go dancing in the streets or something. For society, it's hard to know. Those of us who have a taste for technology and science will be thrilled, and it will revolutionize our thinking. But when I look at the average person and their lack of connection to the microbial world we have here, I'm worried that they either won't care or that they'll be afraid of it. Because, through marketing and other things, many people are left with the idea that all microorganisms are pathogens and are going to kill us, that they're all germs, so to speak, and health hazards. I think as we move into the era of medicine where we begin to understand that our own microbiome is our ecosystem individually and that our health depends on that, I think maybe the favorability rating of microbes might go up. And so maybe there'll be more of a connection there. So I hope people are not very afraid. And I think it's our job in the business, so to speak, to make sure that we use all possible due diligence to make sure that we don't present any kind of hazard, either real or imagined, to our populace here.
And movies like this definitely don't help the case with that perception.
It's awful. And besides, the little starfish thing was so cute. I wanted to love it.
Oh, and it was named Calvin, which was even more adorable.
So precious. And then they turned it into a monster. And I'm like, is this an allegory for, raising children and having them become teenagers?
Maybe. Well, thank you so much for joining Norm and I today.
Oh, it was just great fun. I may someday watch it again. I don't know.
So we talked a lot about all of the different weird aspects of biology and everything in the context of this film, Life. And that was, I don't know, there's so much to unpack. And there was so much information she gave us about, how it's actually kind of difficult to even determine what is and isn't life.
Right, right. And in the film, so much of that is just taken for granted, right? The luck they have in this small sample of dirt that they would find one microbe that then they could stimulate and work out. Like, that is just the conceit, I guess, of the film. Listening to Penny talk about the firewalls and all the considerations, I thought the film, I was impressed by, like, okay, they had thought firewall 1 and 2 and 3. They really didn't think through all of that. Yeah. And it made me wish that it was, Penny on that station in that CDC role, because that would actually give us more tension. Right?
Yeah.
Like, more concerns about how not only we could be infected, but we would be affecting the life.
Yes, yeah. A notable thing she said was, "Don't lick the microbes." Yeah.
Yeah. She would not let Ryan Reynolds in to that hatch into that makeshift laboratory.
Yeah, with a flamethrower. Yeah, no, it would totally not. So, the thing we asked her at the very end was like, how would it change her personally, and how she thought it might change society if we discovered life either previously existing or currently existing on Mars. For me, I think it would really make me just that much more insatiable as a space geek, that I would want to just start saying, all right, let's start sending rockets everywhere, because there's going to be life so many places if we found it in our solar system.
Mm-hmm. It's a version of the conversation we've had with our past guests, and everyone's had kind of a different response, although they share that sense of it be life-changing for them in their field. In the past, our guests have talked about intelligent life with communications. Here we're talking about potentially direct evidence, but it's on that small scale. And I totally agree, where I feel like, on Twitter, on our echo chamber of science communicators, people would be stoked. But I have no idea what someone in the middle of America or on the other side of the world, whether they would care that we found one cell of biological life.
Yeah, I mean, for me, this is a question that I'd want to pose to everyone listening, is really, if we found microbial life on Mars, how would you, assuming many of the people who are listening are geeks about microbes and space and everything, how would you try and convince other people that this is a really exciting find? Like, what would be your story? Like, what would you try and tell people of, like, why this is so exciting and life-changing? I'd like to hear people's comments both on YouTube and for people listening in on the Tested site. And you can also reply us at Ariel Waldman and at nchan on Twitter.
Yeah, and once again, thank you for listening. We'll be back next time with another movie or trope in the space pop culture intersection, and we'll talk to you then.